Thanks so much for the introduction, Dan. Good morning, everyone, and thanks to all the organizers from JGI for giving me this really incredible opportunity to explain my research to all of you at once. As Dan mentioned, I'm a PhD student at UC Davis in Doug Cook's lab, and the lab historically is focused on dissecting the genetic mechanisms that underpin something called symbiotic nitrogen fixation, which I'll explain a little bit more about in a moment. But lately, the lab has started focusing specifically on the crop legume chickpea, and that's what I'll be talking about. So nitrogen is reduced nitrogen is essential for all organisms for assimilation into biological macromolecules, in particular amino acids and nucleic acids, and for plants also for chlorophyll, which, as everyone knows, is what drives photosynthesis. So even though reduced nitrogen is essential for all organisms, only certain members of certain bacteria and archaea have actually evolved to take atmospheric nitrogen, which is the most abundant element on Earth, or in the Earth's atmosphere, take atmospheric nitrogen and convert it to ammonia, which can be then assimilated. So for plants typically get reduced nitrogen in mineral form from the soil. And as a result, in agricultural systems, nitrogen is often the most limiting nutrient for growth. So more nitrogen will do more to improve yields than any other factor. And so in industrial agriculture, to get around this limitation, nitrogen is usually added in the form of synthetic fertilizer that's produced through the industrial Haber-Bosch process. And as I think it was Jonathan Lynch who mentioned yesterday, the Haber-Bosch process, which produces nitrogen fertilizer, is the single largest source of greenhouse gas emissions from the agricultural sector worldwide. So one very important alternative for nitrogen fertilizer in agricultural systems and for plants in natural ecosystems is something called symbiotic nitrogen fixation, where plants from the family Fabaceae, the legume family, which includes soybeans, beans, lentils, peas, chickpea, a number of other crops, as well as, of course, a host of wild species. These plants have evolved a symbiosis with certain free-living soil bacteria that are capable of nitrogen fixation when in association with the host plant. And the symbiosis happens in these little root-derived structures called nodules here, and I'll explain a little bit more about those in a moment. And so because of this symbiosis, legumes play a vital role in global agriculture, and particularly in traditional agricultural systems. So all traditional agricultural systems before the advent of nitrogen fertilizer in this century have relied on legumes to provide or to maintain soil fertility. And so this is still, so not only does the symbiosis have an important environmental role in offsetting the use of synthetic nitrogen fertilizer, but it's still essential in a lot of parts of the world today where nitrogen is a limiting nutrient, and in parts of sub-Saharan Africa in particular where synthetic fertilizer nitrogen is not available often. And so a few more details about how the symbiosis works biologically. Root nodule development is a highly intricate process that involves detailed chemical signaling on the part of both the free-living bacterial symbiont in the soil and the plant host in order to form a root nodule. And because of this level of complexity in the signaling pathway, the symbiosis is highly specific, but this specificity varies across the diversity of the symbiosis. So what that means is on the left in this slide, you see a phylogenetic tree of legumes in the family Fabaceae, or all plants in the family Fabaceae, which is quite a diverse plant family. And then similarly, on the bacterial side, ability to form symbiosis with legumes is actually polyphyletically distributed among 12 bacterial genera. So in some cases, you have a specific legume species that partners with multiple specific bacterial species. So for example, the crop legume soybean is notoriously promiscuous, partnering with specific bacteria from multiple genera, and oftentimes whatever's in the soil, or it'll prefer whatever's in the soil to whatever farmers add at sowing. And similarly, you have certain bacterial species or even individual strains that can form root nodules on a wide range of hosts. And then in other cases, you can say the partnership is a little bit more monogamous, with a specific legume taxon partnering with a specific bacterial taxon. So the essential question of my PhD thesis has been to try to define this level of specificity for the crop legume chickpea, and looking over the crop's full geographic range and using whole genome sequencing to describe in immense detail which bacteria are partnering with chickpea where in the world, and what are the factors that influence that partnership. Is it the plant genotype is selecting different bacteria, the bacteria are adapted to different environments, that sort of thing. And so this is useful agriculturally because, as I mentioned previously with the example of soybean, in modern legume cropping systems, farmers will often add specific bacterial inoculants to try to enhance nitrogen fixation, strains that are shown to perform ideally, at least under lab conditions. And so one question is trying to understand how specific strains will perform under different situations. So if we find a strain that works really well from Turkey, where the crop originated, will it work just as well for farmers in Africa? But this system also turns out to be really useful in a number of ways for probing basic questions of bacterial biogeography and population genetics. So biogeography is the study of how individual organisms vary in their distribution in space and time, and also ecosystems. And so to give you an example, we know where chickpea first originated, which is what's now southeastern Turkey. We know its wild relatives and their distribution, and we know how, once those wild plants were domesticated, how the domesticates were distributed first to the Indian subcontinent around 6,000 years ago, after spreading through the Middle East, then parts of Africa around 3,000 to 4,000 years ago, and more recently to Western countries where it's been grown often with specific bacterial inoculants. So there's no sort of there's very few microbial taxa where we have a similar level of understanding, and the reason is until the advent of whole genome sequencing, it was very difficult to describe bacterial taxa with sufficient resolution to make these same sorts of claims. And it's also difficult to sample in such a way where we get a good geographic representation. So here, because we have that information about the plant host, we can ask questions about the biogeography of the symbiont. So just basically, the way this worked is I collected chickpea root nodule samples from all those sites pictured on the last slide, either myself or in collaboration with colleagues who went into the field themselves, and then have extracted DNA either from bacterial isolates cultured from the root nodule samples or just DNA that was extracted directly from the root nodules, performed whole genome shotgun sequencing using the Illumina platform to get sort of crude assemblies that give me enough information for phylogenetics to say which bacteria occur where, and also to look at and to do comparative genomics for gene content, that sort of thing. And then more recently using PAC biosequencing to get full single molecule or single chromosome, full chromosome assemblies that can give me more information about the the genome structure. So the most essential result from this work so far is pictured in this slide, which is just, you can say, species phylogeny showing which bacterial taxa are forming root nodules on chickpea everywhere in the world that we've sampled. And so the first big conclusion from this is, again, there are multiple bacterial genera that form root nodules on legumes broadly. Chickpea in particular, everywhere we've looked, forms root nodules with bacteria of the genus Mesorhizobium. But within that genus, it's actually quite broad, or it's a symbiotic range. So there are multiple bacterial species within the genus Mesorhizobium, up to, depending on how you define it, 15 or 20 species. And there are some interesting biogeographic patterns already of which species occur where. Excuse me. And I'll get into some of that in a little bit more detail. But the other thing that pops out quite quickly from looking at the genomes is that even though on a species level, chickpea is nodulating with lineages that have been divergent evolutionarily and on different continents for millions of years, all of these bacteria, when you look at genes involved in symbiosis, those genes are almost identical. And the clear hypothesis to explain this is that they're acquiring symbiosis genes through horizontal transfer, and it's been shown in other Mesorhizobium systems in particular. This happens through the transfer of about 500,000 base pair region of the bacterial chromosome called the Symbiosis Island. And so this is why we chose to do pack biosequencing for a certain phylogenetic subset of the strains that we have, of the diversity we had observed. So this plot is showing the reference genome for one strain that was isolated from Turkey, looking at nucleotide diversity across the populations, the bacterial populations throughout Turkey, across the bacterial genome. And what you see is this region highlighted here that shows much higher levels of conservation. And when you look at multiple bacterial genomes, what you find is that same region is conserved in synteny and in levels of conservation across all of the taxa we find. So that confirms the Symbiosis Island hypothesis. So again, this has relevance for agriculture, determining if we find one strain that works really well, can we use that strain in a certain area that's either grown chickpea before or hasn't, and how will it interact with the bacterial strains that are already present in the soil if they can all acquire the Symbiosis Island. But again, this also has relevance for more basic principles of bacterial biogeography. So one question I've been focusing on in particular lately, like in the last few weeks, so bear with me if some of this is still kind of clunky, is there enough signal in the Symbiosis Island to try to track its movement through bacterial populations in the soil, something that hasn't been done in too many bacterial systems before. So essentially, I'll show sort of a truncated version of how I've approached this question. This is another species phylogeny, but just focusing on the 3 Mesorhizobium species that we observe on the wild relatives of chickpea and their native range in southeastern Turkey. And so to go through it quickly, you see that there's 3 Mesorhizobium species that actually have distinct host ranges, and they also have distinct ecological ranges. So there's 2 different chickpea species that occur in southeastern Turkey that are related to the crop. So one Mesorhizobium species nodulates one of the wild plants, and then the other 2 nodulate the other host. And they have distinct ecological ranges. So the question is, how is the Symbiosis Island moving through these populations? Is it corresponding to geography, like 2 strains from different species at the same site will have a similar Symbiosis Island, or is it corresponding to the species? And so what we find, at least in the case of Turkey, pretty strikingly, is that it corresponds to phylogeny. So this is the same strains, but a phylogeny constructed from all of the genes from the Symbiosis Island. And what you see quite clearly is it corresponds just with how the strains are related to each other. So for example, here you have 2 strains that are from the same site, pictured here, but from different species. And conversely, you have 2 strains from different sites that are where the Symbiosis Island is much more closely related because they're from the same species. Now, so what I'm trying to do at this point is expand this analysis not just to these 3 species from one region, but to ask the question of how does this interaction play out across the world? And that analysis at this point looks like this, which I won't make you stare too long at this nauseating pinwheel. Instead, I'll show you more pictures of root nodules, and just to say that one of my goals, my background nominally is in plant pathology, and one of my goals in coming to this conference was to interact with people who think more full-time about population genetics and bacterial populations. So if you have suggestions on how I can apply quantitative analyses to answer these same types of questions given my data set, please, I'd love to collaborate or at least discuss. And so that's sort of the end of my data. Being an international project, there's way too many people for me to thank directly, but this is at least all their names. And hopefully I have time for a couple of questions.
Thanks, Alex. Question over there. Do you have a microphone nearby? I've got it.
Thank you for an interesting talk. So did you, the Symbiosis Island transfers through a phenylalanine tRNA gene. I was just wondering if you considered looking at metagenomes and looking at that marker to look at the transfer through of the Symbiosis Island in the native rhizobia or in the bacteria that are in the rhizosphere.
Well, so I do have, about half of my whole genome sequences from Illumina are from metagenomes from the plant, or from the plant tissue. And unfortunately, in those cases, I can't, often they're not assembling well enough to be able to look at those kinds of relationships. I haven't looked specifically at where, now that I know from the packed biosequences what the Symbiosis Island looks like, I might be able to look specifically at where it's inserting. But what I can say, So this is showing the Symbiosis Island in the 14 genomes that I've constructed from PacBio. It's a pretty busy slide, but what you can see is actually, so for 10 of these 14 sequences I have, this is the Symbiosis Island where you can see it's highly conserved and synthetic. It's inserting into the same site highlighted here in red. And this is the this is the ribosomal operon, and it's inserting in the tRNA that you mentioned. But in 4 cases, actually, it's not inserting into the same site. It seems to be inserting sort of randomly in the genome. So I haven't figured out yet how to apply that to the full population level sampling, but it's yeah, thanks for bringing that up.
We have one more question in the front.
Hi. So you mentioned that it seemed like it was tracking with the phylogeny, the Symbiosis Island, and you also said that it seems like you're supporting a horizontal gene transfer event. Does that mean that the horizontal gene transfer occurred like in an ancient time, and then it followed vertical transmission from there, or well, what's the hypothesis? It's supporting 2 hypotheses.
Yeah, so that's an excellent question, and one that, again, I think my data is well equipped to address, and I haven't completed the analyses yet, but I can give you kind of one piece of anecdotal evidence. So this is showing just a whole genome alignment from 2 of my strains of the 14 that I have whole genomes for, and what you can see is, so this is comparing to 2 other species where the Symbiosis Island is actually broken up by a region in the middle that, even though it's all transferring together, it seems to be diverging pretty rapidly. But in this case, these 2 strains that I just picked randomly happen to have an almost complete Symbiosis Island that's completely conserved. So it's clear that those 2 strains share a much more close, or much more recent intermediary. And interestingly, they're from different sites about a few hundred miles apart and from different species. So I'm expecting, again, if I squint long enough at this slide, that I'll be able to infer those types of patterns, or again, if someone has a better suggestion. So I think the data is set up to answer those questions, but I haven't gotten to it yet.
I have a quick question. Can you make a decent hummus from the wild relatives?
They're pretty small seeded and highly precious because the seeds shatter, so they're hard to get. So, but they do
You'd
taste good. I can say that.
Eat them.
Yeah, exactly.
Okay.
But my favorite way to make hummus is if you can find fresh green chickpeas, like sugar snap peas, essentially. That's really good. You can just grind them up.
Thanks.